Hello today. So a small video on mutations. Look, what is a mutation? When the sequence of nitrogenous bases on the DNA is changed, it will result in an abnormal genotype, and the RNA formed will be abnormal as well, and this abnormal RNA will code for a different type of an amino acid, and the protein formed will be abnormal, and this is a mutation. The phenotype will be damaged as well. While coming to mutations, there are four broad types. The first one is a point mutation. In a point mutation, what happens is that a single nitrogenous base is replaced by another. It can be of two types depending upon the type of the nitrogenous base being replaced. In transition, a purine is replaced with a purine, or a pyrimidine is replaced with a pyrimidine. While in a transversion variant, a purine is replaced with pyrimidine, while pyrimidine can be replaced with a purine. It can be having three possible outcomes, one of the three. A silent mutation. If the mutated codon codes for the same amino acid, as you know, the possible codons are 64 and the natural or the standard amino acids are 20 in number. More than one codon can code for the same, amino acid. UCA codes for serine. If it becomes UCU, UCG, or even UCC, the amino acid will be serine. So it would be a silent mutation. The genetical code or the genotype will be abnormal, but the phenotype will be normal. Now, a missense mutation. If the mutated codon codes for a different amino acid. Sickle cell anemia, GAG codes for glutamic acid. Mutated into GTG codes for valine. Other examples: Alzheimer's disease, amyotrophic lateral sclerosis, HBC disease, HBD disease, HBE disease. All of these are missense mutations. Then a nonsense mutation. If the codon becomes, if the mutated codon, in fact, becomes a stop codon, UAA, UAG, or even UGA. For example, Duchenne muscular dystrophy, beta thalassemia, cystic fibrosis, the Rett syndrome, and even Herder syndrome, the famous examples. Now we come to the next type. This is the frame shift mutations. If an extra nitrogenous base is added or a single one is removed, all of the next codons will be abnormal or damaged. Tay-Sachs disease, Crohn's disease, and in rare cases of cystic fibrosis as well. Then splice site mutation. In this, a part of intron remains embedded inside the RNA, which should not be there. Beta thalassemia, Gaucher's disease, Marfan syndrome, TTP, and some rare cancers as well as in dementia. Then we come to the fourth one, trinucleotide repeat mutations. A codon is amplified multiple times and the same amino acid comes again and again and again and again. Huntington's disease, myotonic dystrophy, spinocerebellar ataxia, even Fragile X syndrome. I hope this summarizes all of the mutations. Thank you so much.